We're going to start by introducing a new approach to medicine, and we'll begin by reviewing briefly the history of medicine and the introduction of reductionist thinking into the scientific process. We'll talk a little bit about epistemology, which is the science of how we know what we know. The limits of reductionism. Will differentiate integrative medicine from integrating medicine and come to some basic conclusions. Currently, we're in a state of what is referred to as naive reductionism, which reductionism by itself is the tendency introduced by René Descartes to reduce complex subjects to their simplest elements. And when Descartes talked about, he introduced this idea into scientific reasoning, he explained that he would build back up to the complex idea once he had broken it down to its simplest elements of activity. But the concept of naive reductionism takes Descartes' idea of reducing complex subjects to simpler elements into elements which no longer represent or reflect the whole in any demonstrable way. And really, this tendency has come through a series of observations over the last, say, 120 years, starting with Darwinian evolutionary thought, and then the discovery of DNA, and then the entry into molecular biology. As we go further and further away from biologic systems into non-living complex systems, in other words, the world of chemistry and then the world of physics, we start to leave the emergent properties which are expressed in living organisms. And what we look at is no longer reflective of the actual whole complex organism that we call life. And this is naive reductionism. So we see here a photograph of a bird that's living and a dead and partially dissected bird. And you see the two are not equivalent. And in the same way, all of these in vitro studies or high throughput metabolomic assays that are being run do not reflect the actual functioning of the trillions of cells in a complex living system working all together. Even if you engage in a systems view of a single cell and you look at all the complex metabolomic or proteomic activity or genomic activity, that is only one cell. And unless you're studying a unicellular organism, you have no idea how that single cell is interacting with its local cells, the regional cells, and the global organism as a whole. It provides some information, but for clinicians, it is not the information that becomes relevant and applicable to the care of your patients. How did we get to this point of naive reductionism, which is quite in force right now in academic circles around the world? This is the basis of funding. Major research projects are all based on this concept of reductionist thinking and naive reductionism. This is not only not a fringe, this is the foundation of modern science. Well, we started with a holistic approach. Before the time of Descartes and the Age of Reason in Europe, we had an approach to science which was rooted in philosophy and cosmology, and which saw, in the case of human beings as a whole, and saw the interaction of the human being with their environment. I think some of the shortcomings that existed at that time were that there was a lot of dogmatic thinking in scientific circles. So once Aristotle had stated something, or Ptolemy had stated something, or some great sage had stated something or written something in a book, it could be repeated for 1,000 years and nobody would ever try to verify it or contradict it. So there was definitely a tendency to be very sclerotic and not engage in original thinking or to trust your own observations. And so in the light of that, people like Descartes or Francis Bacon or other thinkers started saying, well, let's start to use reason instead of just repeating what somebody told us. Let's observe, let's test, let's verify. And these were really profound concepts and important ones. And what we will present this weekend in Endobiogeny is based on this very concept of analytical thinking, of testing, of verifying, of creating objective means of demonstrating to other people what an individual is observing. So these ideas were truly revolutionary and important. And Descartes' idea of reducing complex systems to their simplest elements was also a very important idea. Because not understanding the parts that make up the whole can lead to a lot of confusion about what is cause, what is mechanism, and what is effect. It was, in our opinion, it was really the tendency to start to get down to cellular and then subcellular activity which started to abstract the notion of biologic life and reduce the complexity of living organisms to simply a collection of atomic and chemical phenomena. And it is this leap into this naive reductionism which has created, in our opinion, a barrier to advancing medical science and personalizing healthcare for the needs of each individual. In the 17th century, in the 1600s, there was a number of important ideas which came into development. And Laplace and Newton and Descartes were the main thinkers who helped shift this idea on the continent. And they believed that the universe was characterized by control, order, and predictability. And because of these concepts of being able to describe complex events through mathematics, and believing that the God of the universe was a God of control and order, and that if you could know all the determinants, you could predict everything with certainty, became the basis for then later what we call Newtonian mechanics or Newtonian physics, and the foundation for clinical studies and scientific enterprises. And so part of this philosophical shift was to shift the questioning from why things happen to how things happen, from cause to mechanism, and from the whole to the part. So again, at the cusp of this revolution in thinking, there was an effort to understand the parts, but to return to the whole. But over the last 500 years, we no longer look at the whole, we look only at the parts. And the parts that we can reduce a living organism to become smaller and smaller. In the 19th century, with the advancements in optics and the more powerful microscopes and ways to fix tissues, we started to get into histologic studies and experimental physiology, and the concept of the interior milieu, coined by Claude Bernard. We had Pasteur's germ theory, we had Koch's experiments in injecting microorganisms into animals and reproducing infectious disease. We had analytical blood chemistry that came out of analytical chemistry. And then we have the ability to establish complex quantitative methods of analysis. And so these advances in technology further reinforced this mechanistic and reductionist and quantitative approach to clinical science. The whole foundation of what we do when we measure a blood count, when we take a tissue biopsy to see if there's hepatic cirrhosis or to characterize a cancer, is based on these types of technologies and these concepts. But even in the 19th century, there were people who were opposing these steps into further reductionism. The great German neurologist and pathologist Virchow himself strongly opposed Pasteur's theory, his germ theory, because Virchow said he had observed that in thousands of histologic specimens of cadavers, he found microorganisms that were not the cause of death of the patient. And he had understood that microorganisms can live inside of other more complex living organisms without causing disease. And Virchow had appreciated the fact that there needed to be a terrain which allowed those microorganisms to become pathogenic and not just colonizing. But again, what happened is, as science progressed in the 20th century, we had a number of important factors occur. One, we had the advancement of molecular biology, endocrinology, cellular biology, DNA, biophysics, and now the omics and big data science. And then, more importantly, we had the development of pharmaceutical companies and the translation of analytical chemistry into the modern concept of a medication. And then after World War II, starting in the United States and then in other countries, we had a major initiative where governments and industry and academic institutions formed a triumvirate and created a specific agenda or approach to studying medicine and funding, which then took us deeper and deeper into this way of thinking. So, we are all trained in this system. And being a part of this reductionist system, we say, well, what's wrong with this system? It's produced life-saving medications. It's allowed us to have heart-lung bypass for cardiac surgery and transplantation, and mechanical ventilation, and diabetes medications, and it's allowed us to understand how medicinal plants work and the pharmacokinetics and pharmacodynamics. It's allowed us to explore our world. It's allowed us to develop nanotechnology, which one day might assist us in some way in medical care. What is wrong with this system? Well, we're not saying that the system is bankrupt. And we're not saying that the system lacks merit. Our approach in endobiogeny is not to replace or negate what has come before us. It is to enrich and deepen it by stating in no uncertain terms that life and the expression of life is a complex modality of expression where each human being is related to other living organisms, both within itself, around itself, and with nature and the earth around us. And it is this approach of looking at complex living organisms and looking at interrelatedness and the integration of these parts to the wholes, which will allow us to create a more personalized form of medicine and an approach to medicine which is respectful of every living being around us. The current system, as we know it, says that a symptom is a loss of control and order. Because remember, these 17th century thinkers said that the universe is predicated on control and order, not on dynamic and complex chaos and interrelatedness. So with this thinking, we say, well, if there's a symptom, there's a loss of control in the body. And the cause is now attributed to faulty genes, because we say a gene makes a protein, and a protein does something in a cell, and the body is just a collection of trillions of cells. So if you have a disease, somewhere along the line there is a genetic problem, and therefore, if you don't have control, you have to reestablish control. And therefore, the approach to treatment that we use is to control the symptoms. And the more controlling the drug is, the more it can suppress a symptom, the more it can control or block or stimulate forcefully some type of physiologic process or biologic activity, the better the treatment is. And so we have developed a system of treatment which is based on taking a single compound with a single mechanism of action at a single site, a single receptor, a single activity in the body. And this has become the foundation of our treatment. Are these treatments valuable? Of course they are. But it is only in the most extreme cases of dysregulation where the organism has lost essentially its buffering capacity and its ability to heal itself that these medications become truly helpful. Long before you would need these types of medications, there's a whole series of other interventions that could be offered to a patient. Lifestyle changes, dietary changes, mineral therapy, the use of medicinal plants. Part of the problem is that, coming back to this concept of analytical chemistry and blood chemistry and histology, is that we only equate disease or dysfunction when there is a massive and significant alteration in histologic integrity. So, put simply, unless liver enzymes are elevated in the blood, we conclude there's nothing wrong with the liver. But the liver does over 500 different things in the body. And if you want to wait until there's so much histologic damage to the liver that you can measure liver enzymes, you will have waited until the body is already in some important state of decompensation. In your own house, you would not wait until a storm to fix a hole in your roof. You would want to make sure that the integrity of your own house is maintained at all times and prevent its degradation before you have to fix it. And yet somehow, with the human body, we wait for something to become seriously wrong before we even acknowledge to the patient that their symptoms are valid. So part of this approach is that it becomes dehumanizing. It denies the patient their own experience of their symptoms. Because when a patient comes to you with symptoms and normal labs and normal imaging studies, you don't say, I'm an incompetent doctor. You say, you are a crazy patient. And your solution is to prescribe them an anxiolytic medication or an antidepressant. And instead of holding that conflict in our minds as clinicians, why can I not understand the origin of this patient's symptoms? We simply invalidate the patient's own experience of their life as a complex living organism. And when we give a medication which forcefully stimulates or blocks some type of biologic or physiologic process, we create a whole series of side effects. Because complex and dynamic organisms always strive to maintain the maximum amount of dynamic activity. Because the more dynamism, the more interrelatedness there is, the greater the degrees of freedom for that organism to operate. Simply put, I mean, for the honorable people here of Lithuania, you know, to live under a repressive and controlling regime that limits your degrees of freedom, of expression, of activity, is something which is contrary to human nature, which is to be free. And the very essence of all complex organisms is to have the maximum degrees of freedom. It is not just at the meta level of human consciousness that we want to be free. Everything wants to be free, to operate in its maximum degree of potential. And the current system of clinical medicine is about limiting degrees of freedom. It is not about encouraging degrees of freedom. So where do we go from here? How can we possibly expand on such a powerful and strong system of reductionist thinking? And again, we're not saying that there's no benefit from this system. There is benefit. Every day people come to us who are severely decompensated, who have uncontrolled diabetes, who have uncontrolled hypertension, who have congestive heart failure, who have glandular disorders. But those patients represent a fraction of the entire population. And if we can recognize when a patient is in a pre-critical state of illness, when we can recognize when the organism is still in a dynamic mode but has a reduced degree of freedom before its expressed symptoms, if we can help other human beings maintain and regain their optimal state of health, that would surely be far better than waiting for them to become decompensated and then trying to intervene. So our approach, which has already been adopted in many other forms or areas of science, is something that we call systems theory. Fritjof Capra in The Turning Point, Science, Society, and the Rising Culture says, living systems are organized in such a way that they form multi-leveled structures, each consisting of subsystems, which are wholes in regard to their parts, and parts with respect to larger wholes. The idea in systems theory, very basically put, is that there are a series of nested hierarchies. In other words, each one of the smallest circles represents a unit. In and of itself, this unit is its own system. But when a series of these units are collected together, they form a new system, which has properties that were not seen in each of the smaller circles. And then all of those collections of larger systems are part of another system, and so on and so on. This is what we mean by a nested hierarchy. Systems within systems within systems. Each one of the smallest circles maintains its own independent level of function, but it is interdependent on the activity of all the other systems. So in other words, if you think about it at a macro scale, when you are in a family, as a son, as a father, as a mother, as a daughter, you maintain your own independent existence, but it is always linked to the activity of the other people in your family. Even if you say, I'm never going to speak to you again, you're still linked to that person, right? You never really cut off those relationships. And it's the same way at every level of existence. Units function on their own, but they function in a way which is dependent and influenced by the activity of the other units. We'll come back to this in more detail later on in the talk. But here I just want to show you that we can have a very static and conceptual understanding of these nested hierarchies in a system, and then we can start to look at how they are connected within each other and to things outside of themselves. And then we can create something like a heat map where the colors in this diagram represent not just the fact that units are connected, but to what degree they're reliant on each other. Because, for example, you can have a connection to something, but infrequently connect to it, and you can have a connection to something and frequently connect to it. So, for example, you have a connection, once again, to your parents, and you may visit your parents every weekend or every holiday or every year, and you may have a connection to a religious site, but only make pilgrimage to it once in your lifetime or twice in your lifetime. So as we start to analyze a system, we not only want to look at what are the parts and how are they connected, but to what degree does one part influence another part? So where do we go from here, specifically within the clinical sciences? Well, we have a molecular and submolecular, a subatomic way of looking at biologic organisms or living organisms. We can look at the molecules within those living organisms. We can look at the DNA and the proteins that they form. We can look at the membrane and look at the cell as a whole. Or we can look at how those cells form tissues. And this here is an image of retinal cells. Or we can look at how those tissues act within the organ as a whole. Or we can look at how that organ interacts with other systems in the body. Or we can look at how all those systems interact with the universe around us. Because as living organisms, we are permanently linked to cosmobiologic phenomenon. Life is expressed through rhythmic activity, fluctuations, which are in essence part of the dynamic nature of every complex system. And living systems are linked to the changes of day and night and to the changes of seasons, which are directly related to the rotation of the Earth, the revolution of the Earth, and the amount of electromagnetic and photonic exposure that we have from the sun. And what links us to these cosmic phenomenon is the eye. And we can go into the eye, and we can go into cellular, and again, DNA and molecular, and quantum physics activity. All of these levels of analysis are valid. Each level tells us something that the other level doesn't tell us. But the concept of complex living organisms is that there is emergent properties, properties that don't exist at one level, but exist at a higher level of function. So again, it is not that we are not interested in scientific study or reducing complex systems or looking at different levels of function. It's just that when we talk about something like a chronobiologic disorder, like insomnia, the inability to regulate our internal rhythms to the external rhythms of our environment, this is not a minor thing. Sleep is so essential for reconstruction and repair of the body, for the functioning of the organs of detoxification, for dreams and reorganization and consolidation of memories, and just for the general quality of health of human beings. And multiple studies have shown that disruptions in chronobiologic function and suppression in melatonin is correlated, not in a causational sense, but correlated to an increased risk of cancer. So for example, nurses who work the night shift for a prolonged period of time have a higher incidence of breast cancer than nurses who work the day shift. And being up at night and being exposed to fluorescent lights and suppressing melatonin when it's meant to be expressed has certain consequences. But we will not be able to understand and treat chronobiologic disorders by analyzing quantum phenomenon, because the human being as a whole is not a quantum phenomenon. Human beings as a whole do not follow the Heisenberg uncertainty principle. We do not have a high degree of probability of being here and somewhere else at the same time. And you can know the location and speed of a whole human being in the way that you can't know that for an elemental particle. So all of it is interesting and important to understand the wavelength, the spectra, photonic impacts. We need to understand this to understand how the whole system of regulation of the pineal gland, of melatonin, etc., occurs in the body. But ultimately, to treat the organism, we need to know more than just that the pineal gland makes melatonin. Because melatonin regulates different organs in the body, and those organs regulate different types of functioning. And we'll allude to that later on in the lecture. So what we are advocating for is a global systems approach, which understands and respects the functioning of the parts in and of themselves, but also in relationship to each other and the global system. Medicine is a cultural product. Medicine in all science is no different than going to the theater to see a play by Chekhov, or listening to Beethoven's Ninth Symphony, or eating an artfully prepared meal. They are all cultural phenomenon. When people organize together, they start to share concepts and beliefs, and they create a language. And the words that are contained within a language express the concepts that a people are capable of possessing. If you don't have a word for it, you don't have the concept. And when you have a concept and you don't have a word, you either make up a new word or you borrow a word from another culture that had that concept and shared it with you. But we really need to understand that it's not science is here and the humanities is here and never the twain shall meet. They are all part of the same cultural and human endeavor to understand ourselves in the world. And the reason that what we are interested in studying changes over time is because our culture changes. We only can study what we can conceive of. If you look at the early studies in ethography, in animal studies, in animal societies, you see that the 19th century and early 20th century ethographers, which studied, for example, primate behavior, saw and described primate behavior as a projection of simple more simplistic tribal and hunter-gatherer types of human societies. So we talked about alpha males, and they talked about harems, and they talked about these very rigid control structures and power structures where one male chimp was on top and everyone else had to listen to it. But lo and behold, when a woman like Jane Goodall started studying chimpanzee behavior, all of a sudden people started noticing cooperative behavior, the use of tools, and a whole different structure to the society which was not seen before. In the clinical sciences, up until the 1960s, in the United States, women were not studied, children were not studied, and people of color were not studied. Every single clinical trial of a medication or a surgical intervention was essentially done on white men. It was no different than the Greek concept of democracy, of being a wealthy, white, landowning male. It was just a projection of, in our culture, of who is important and who is worthy of being studied. So science and medicine is a cultural endeavor. Those of you like Daugiau who've had the good fortune of going through a surgical residency know that it is the closest thing in civilian life that you can get to being in the military. And that's not by accident. The entire order and structure of contemporary medical training is based on Napoleonic military medicine. Prior to the time of Napoleon, surgeons were considered to be in quite low esteem in general. And it was the physicians, the medical doctors, who were held in high esteem. And Napoleon could not get enough surgeons even, or medical doctors, to travel with his army as he expanded eastward. And so he simply brought in barbers who were also allowed to cut and operate. And from that, the prominence of surgery as the supreme medical science started. And the whole basis of 19th century and then 20th century medical education was based on this military type of model. There's a chief, and you are put through a punishing series of trials and sleep deprivation and overworked, and you're yelled at and you're humiliated until you learn to fit into the system. Because this is the society that we lived in. And as society changes, as more women enter medicine, as you know in Lithuania, I think the majority of medical students are now women. The nature of medicine is changing, the nature of training. I can tell you that in the United States in the last 20 or 30 years, the whole basis of how you even select somebody to enter into medical school has changed. Before, it was only based on your grades. And now we look for people who have a richness and depth to their character. One of the first things that I learned in medical school was how to care about the patient so I could care for the patient. And this was such a radical shift in what medical education was to mean. I specifically had classes on how to interview a patient, how to listen to a patient, how to present bad news to a patient. Because there was a shift in the consciousness of our culture of what it meant to be healthy, what it meant to be sick, and what it meant to be a physician. So science and medicine is a cultural endeavor. And when a fish never thinks about the water that it swims in, it can't understand why it swims in that water. So we come to this concept of epistemology. And epistemology is the study of how we know what we know and the ways of knowing. And even though this is a course on medicine, it is really a course on so much more. And every good scientific endeavor has to start with philosophy. Albert Einstein said, "Science without epistemology is primitive and muddled." If we are to come to a deeper understanding of what it means to be alive, what a complex living system is, and how we can interact with another human being as a living organism in a sensitive and respectful way, we need to understand what we understand and how we came to hold those beliefs. Otherwise, we cannot get outside of that belief system. So in general, we can say that, according to this Venn diagram, there is this abstract concept of the truth, and then there is our beliefs and what we have the capacity to know. And where the truth and our understanding of the truth overlap, we get knowledge. And this is how we start to know. Now, broadly speaking, we can say that there are 5 different ways of knowing: a traditional approach, an empirical approach, a rational approach, an intuitive approach, and a synthetic approach. And as is true with any type of complex system, these ways of knowing are imbricated and intercalated. They are nested within each other and constantly related to each other. So, for example, when we talk about traditional knowledge refers to knowledge or a way of doing things because that's the way it's been done. And the belief in the traditional approach to knowing is that if it was not worth doing, people would not have continued to do it. And that the continuation of a way of doing things and a knowledge base is a justification of its wisdom. And that wisdom retains the good and discards the bad. And therefore, in a traditional way of knowledge, you don't seek proofs. Because the fact that it is done and you see it as being worthy is all the proof you need. This diagram here is an Ayurvedic diagram from the ancient Indian system of knowledge of medicine. And the same thing could be said for, say, traditional Chinese medicine and other ancient and complex methods of medicine. Now, those of us who are trained in, if we go back, in an empirical and rational scientific method, we might find this to be somewhat primitive or amusing. But in truth, 80% of the knowledge that physicians acquire are acquired in your 3 years or your 5 years of training. You will only learn 20% more information over the next 30 or 40 years of training, of practice. Because people are so resistant themselves to learning new things. On average, it takes over 20 years for something which has been definitively proven to be best practice to be implemented by community physicians. So we may think that as rational clinician scientists, we are not subject to traditional knowledge. But you just think about a country like as small as Lithuania, or even a country as big as the United States. The majority of physicians practice within 100 kilometers of where they completed their training, which means they are practicing the way everyone else is practicing. And we have this concept, at least in the United States, called the community standard, which says you should be practicing the way that other doctors in your community are practicing. And if you don't, you're a bad doctor. But this is just reinforcing a traditional way of knowledge without thinking about why do we operate on an enlarged prostate? Why do we inject steroids when a person feels a little sick? Why do we have this level of utilization of some type of therapeutic process? Again, just citing research from the United States, there are completely different rates of prostatectomy or lumpectomies or utilization of MRIs in different regions of the United States. If science is science and medicine is medicine and best practice is best practice, there should be uniform rates of surgical intervention and utilization of diagnostic imaging and prescription patterns. But we don't really practice scientific medicine. We practice traditional scientific medicine, which is you look at the people around you, you look at what somebody taught you, and that's how you practice. Empirical knowledge is knowledge which is obtained by observation. And empiricism has some important value because the personal experience of each physician or clinician is valuable and it should be respected. Because people naturally tend to attract to themselves certain types of patients, or people tend to have an interest in certain types of pathophysiologic states. And so people naturally gain experience with particular types of conditions. The challenges with an empirical approach is that we do not practice, as William James said, radical empiricism. Radical empiricism, as William James, the founder of modern psychology, described it, is an empirical method where whatever you see, you accept. And if it displaces what you believed before, you let go of what you believed before. The problem is very few people are bold enough to practice radical empiricism. And, you know, we have a saying that says that, that success has many fathers, but failure is a bastard, right? Nobody wants to admit failure or take credit for failure, but everybody wants to take credit for success. And in our own attempt to protect ourselves and our own concept of how good we are as a clinician, we tend to hold on to those amazing cases and results that we had, and we tend to forget about the patients who didn't respond to something that we were just so sure they were going to respond to. So empiricism has its value because we want to respect every person's own understanding and the conclusions that they came to, and honor and take advantage of what they have experienced and observed. But each of us, as we embark on an empirical way of knowing, need to be aware of to what degree we are engaged in traditional knowing and excluding anything which doesn't verify what we already believed. Rationalism is the basis of the modern scientific method. And again, rationalism offers a truly profound and important shift in human consciousness. It states that knowledge can be objectively and systematically determined. And the idea of taking a systematic approach and taking an objective approach is a truly important one because it sets aside the traditional and empirical approach at first and says, let's start assuming that we know nothing. And the goal of the scientific approach, especially when you form a hypothesis, is not to prove yourself right, but to make sure that you're not wrong. That is what the null hypothesis always says. And that is a profound way of thinking. It is very humbling, and it comes from a deep place of humility. If you practice a rational approach to science in this fullest sense. Because all you can really come to a conclusion about is to say, there's a 95% probability that I'm not wrong. That is very different than saying, I'm right, this is the way it is. And, you know, this is part, again, of our epistemologic struggle as human beings is because in our popular literature and even amongst ourselves, we refer to science as almost this God, this oracle. Science says, scientists discovered. And when you want to make an argument, you say science says this, and therefore you can't argue with me anymore because science said it. And this is not in the spirit of a rationalist approach to knowledge. Just like when we look in the field of astrobiology, which has many different levels, but one of them is the search for other living systems throughout the universe. And when people engaged in a scientific field of thought say, there's no life in the universe except on Earth, or the Earth is special because human beings are able to do this and this and that with knowledge and information. This is a very arrogant and really simplistic approach. It is not in the spirit of science to say that. It would be much more accurate to say that based on our concepts of what is a living system, and based on the searches that we've done so far, we are the only planet that has living systems that we know about. But that's not a very sexy or exciting thing, and it doesn't make a great headline. But we all can fall prey to this way of thinking. But again, to the degree that we can maintain a rationalist approach and be systematic and thorough in our investigations, this is an important and valuable approach to knowledge. But where does a rational scientist get his ideas to form a hypothesis? It is through empirical observation. And how does somebody observe something and come to some initial ideas? It's based on a traditional form of knowledge. Because you can only, it's not that seeing is believing. It's believing is seeing. Because if you can't believe something, you can't see it. And so even to be a rational scientist, you are always within an empirical and traditionalist approach. And that's why I was saying earlier that these ideas are nested within each other. You can never really escape one for the other. And according to the theory of endobiogeny, you shouldn't try to escape. Each of these forms of knowledge has an intrinsic value, and it brings a level of understanding which cannot be obtained by the other ways of knowledge. But what is important is for us to know which method of knowledge we're using. We have to know about our knowing. Because when we don't, we're no better than an automaton. We're no better than a robot. So some of the benefits of the modern rational scientific method and a quantitative approach to measuring everything is that we can communicate information with clarity and precision. When we have precise instruments, we can measure things precisely, we can convey our results to other scientists, and we can say, why don't you prove it for yourself so you can become more certain? This is much better than saying, I'm the chief of the department and I say this is how we stent, and that's the final discussion. Because again, it limits the degrees of freedom that each individual has to observe, to come to conclusions, to be innovative, to be creative, and to expand human knowledge. But there's a number of shortcomings to this method as we currently use it. The scientific method has gone far beyond a rationalist approach to being, as we mentioned earlier, a form of naive reductionism. If the current success rate for developing a drug and correct me if I'm wrong, Professor, is 15%. I actually reviewed the literature over about 2 decades, and I looked at the number of new chemical entities, as it's called, and the number of drugs which come to market. For every 100 new chemicals that are discovered, 15 of them become drugs that make it to market. That means that the reductionist approach of finding a chemical, testing it on some subcellular process, and then on cells, and then in animals, and then in phase 1, phase 2, and phase 3 clinical trials, has only a 15% success rate. If you turn that around, modern clinical science has an 85% failure rate. 85% failure rate in making a medication that is actually more beneficial than harmful for a human being. If our farmers had an 85% crop failure rate, if Apple computers had an 85% failure rate, if Boeing made planes which crashed 85% of the time and couldn't stay in the air, I think we'd be a little dissatisfied. But somehow, we find ourselves in a system which is so certain of its approach to understanding complex living organisms that it accepts an 85% failure rate as a sign of success. And constantly we see in medical journals, in popular journals, scientists discover new drug promises hope. But 85% of the time, there is no hope. There is no light at the end of the tunnel. And these are just the drugs that make it to market past phase 3 trials. And you know how many scientific studies which demonstrated harm were suppressed by pharmaceutical companies for decades. And it is only in post-market analysis that we see the true harm of many drugs. Yes, there are drugs being produced, but the side effects and the failure rates. If this concept of naive reductionism truly reflected the functioning of the universe and of complex systems, we wouldn't have an 85% failure rate. But it doesn't. Because once you give somebody a medication, you're not treating a cell. And you're not treating an animal who you created a disease in 3 weeks ago. You're treating a human being who has been in 20 years of a precritical state and 10 years of mild symptomatology and 3 years of frank dysfunction before they even saw their physician. And it is the ability to maintain this level of disconnect and failure and colossal waste of money and to say we're right and this is the greatest method of knowing that has ever been developed in the history of the universe, when we don't even know if other beings exist and how they think, is truly problematic. And what we argue for is not the abandonment of the scientific method, but a return to the true spirit of a rational and open-minded approach to medicine, which respects the true complexity and dynamism of a living organism and holds this complexity and dynamic nature as the foundation of how we ask the questions that we want to ask. You see, answers are easy to find. Good questions are hard to find because the question you ask determines the answer you get. And if we don't know how to ask the right questions, we can't get the right answers. There's a fourth way of knowing called intuition. And we might think, what place does intuition have in science? Intuition is for poets. Intuition is for artists. Intuitions are for eccentric scientists. I mean, maybe someone like Tesla you think about. But good scientists are rational, great scientists are intuitive. And the history of science has demonstrated this over and over again. Does anybody know what this image is here on your right? It's called Ouroboros. It is the snake eating its own tail. Do you know why I put this image up here? That could be one meaning. Because I want snakes to eat themselves. No, because the foundation of modern analytical chemistry was because of this image. Kukle, the German-speaking scientist, was trying to understand the structure of the benzene ring, the simple 6-carbon formation. And he was sitting in his armchair, and he was starting to fall asleep. And in that moment between sleep and wakefulness, what we call a hypnagogic state, he saw a vision of snakes biting their own tails and swirling around his eyes. And then he realized that the structure of benzene was a simple 6-carbon ring, where the first carbon was linked again through the last. And there are many other examples of this. The modern engine that we use came to Tesla, Nikola Tesla, in a vision. He was walking down the street and he saw a vision and he drew it in the sand and he instantly understood how to make a more efficient engine. Einstein's special theory of relativity came through intuitive knowledge. Linus Pauling's concept of hemoglobin being deformed in sickle cell anemia came through intuitive knowledge. And on and on and on it goes. Intuition is not just some weird thing that happens to poets dancing around a fire after drinking too much vodka. Intuition is a supra-rational way of knowing. Being rational is as really like slogging through a very dense forest. Because to be rational, you have to keep cutting things away, you have to keep clearing the underbrush, you have to separate, you're confused, you don't know, things are a tangled mess. And interestingly, in Farsi, in the Persian language, the word for rational and deductive thinking is halaji, which means to card the wool. You know, when you beat wool to separate all the strands out till you can see each one by itself, this is really what it is like to be a rational thinker. It is a very laborious process. It has its rewards, but it's very laborious. Supra-rational knowledge is a way of grasping the whole and knowing without thinking. It is an instant understanding of the gestalt. But the reason that great scientists are intuitive is because they also have the rational knowledge to prove what they saw in an instant to everyone else. And again, we're not saying that all science should be based on intuition and that, you know, scientists should abandon their laboratories and sit around a circle of oak trees and, you know, invoke perkunis and wait for some idea to come to them. But we're saying that there are other ways of knowing, and a super-rational way of knowing that grasps the whole, which understands the global complex system, is a valuable way of knowing, but only if you have the other forms of knowledge to then make something of it and prove it to other people. And finally, the fifth way is the synthetic way. And the synthetic way honors the first four methods, and it expands the size and depth of knowing. It creates a synthetic approach allows for a holographic way of understanding a problem. It allows you to go through a problem, within a problem, around a problem, look at its relationship in space and time to other similar or different issues. And the synthetic approach is not limited to culture or pre-existing beliefs. It is only limited by our imagination and our ability to grasp the parts and the wholes. In a sense, synthetic thinking is the very essence of what it means to be a complex system, because complex systems are constantly interacting between the parts and the wholes in a dynamic way in which one is never separated from the functioning of the other. And endobiogeny as an approach, as a theory of life, is a synthetic approach to science, which honors the four previous methods. So if we can summarize these 5 ways of knowing in a sentence, we could say that the traditional method is the method where you say it's right because this is how it's always been done. And the empirical says it's right because I've seen the results. And the rational method says it's right because I've proven all other ways as not being right. The intuitive way says it's right because I've experienced its rightness in an instant. And the synthetic approach says it's right because I've intuited, then tested, then empirically observed the results, and then I've reconfirmed it as supporting what has been traditionally done, or I have invalidated what was traditionally done. Now, in the United States, there has been a movement, say, for the last 20 or 30 years in something called integrative medicine. And integrative medicine is something which was really valuable. It is valuable in its time because it was an approach by people within an academic medical environment to knock on the door of naive reductionism and say, there is something called mind, there is a meta-experience called consciousness, there is an influence of emotions and thoughts on the physical functioning of the body, and there's more to illness in the human experience than the simple biochemistry that you can measure through a biopsy or through some type of analytical chemical method. But from the perspective of endobiogeny, what we call integrative medicine has been primarily a syncretic method. And syncretic means adding things onto each other. So, for example, there's sort of this Faustian bargain or this grand bargain, which says, as long as you don't suggest that there's something that can take the place of our pharmaceutical therapies and our surgery, you can talk about it in the academic center. And the idea is that everything you do is right. We just want to add a few things on the periphery where you don't have a treatment. So this is syncretic. It's saying, well, let's add this from Ayurveda, let's add this from Chinese medicine, let's add this from homeopathy, and let's all be one big happy family and not fight. The advantage of this big tent approach, which says, hey, you know, academic medicine, it's big enough for all of us to be here and get along. It's good because it broadens the therapeutic options and the concepts of the current biomedical model. However, it has been limited because of certain political and conceptual restrictions to body-mind interactions, nutrition, and to proving, for example, that meditation is good or effective by looking at changes in telomere length or interleukin activity. This has been sort of the bargain. The idea is that I don't know what you're doing and I don't know what the mechanism is, but if you can prove some type of change in a simple biochemical marker, I just might let you teach my patients how to meditate. But it's as if to say there's no role for meditation, there's no role for hypnosis in clinical diseases unless you can prove to me that you can change interleukins. An integrating approach is one which is synthetic in nature. It's one which doesn't say, I'm going to stick this on top of what you have. It says, let us see how these ideas are related to each other and how one affects another. Let us look at cause, let us look at mechanism, let us look at effect together. The goal of an integrating approach like endobiogeny is to deepen medicine and to broaden it, but not just to broaden it. Because in our opinion, medicine must evolve its thinking to complex systems theory the way that certain aspects of biologic activity and environmental science and, of course, physics has already done, and even economics has already done. And so, in an integrating approach, it can address all areas of clinical medicine. It seeks proofs, and it can also prove its concepts with biomarkers, but not in a simplistic way, in a complex way which understands what is upstream and what is downstream, and differentiates cause from mechanism from effect. So to conclude this lecture before we take our first break, endobiogeny is an integrating and synthetic approach to understanding what life is, how life develops, is organized, and how health is maintained. It is a new vision of human physiology based on complex system theory and looking at the parts and the wholes and how they are integrated and interrelated at all times. It looks at every level of function of a human being within itself: subcellular, cellular, tissue, organ interaction, global systems functioning, and it looks at the interaction of the human being with other human beings and with its environment around it, both on the earth and its relationship to celestial functionings, and the alteration in photonic input from the sun and the moon, as well as electromagnetic activity, ultraviolet, etc. The endobiogenic approach to the theory of life recognizes both physiologic, cognitive, and emotional inputs, and looks for the relationship between cognitive, emotional, and conscious epiphenomenon and neuroendocrine determinants of how we feel and how we think. In other words, there's no psychiatric disorder which is not rooted in some or related to some type of neuroendocrine dysfunction. Finally, endobiogeny is not a replacement or an abandonment of modern science. It is an attempt to bring in complex systems theory from other fields of scientific thought and enrich and deepen medicine and bring it forward into a new humanistic yet scientific approach which places each patient at the center of the evaluation and personalizes the evaluation and the treatment to respect each individual and their own experience of their suffering. So I'd like to thank you at this time. If there's any questions, we'll take them before we start our break in a few minutes. Questions, thoughts? All right, then we have a break scheduled. Oh, actually, we ended a little bit early. So we'll take a break. We'll take a break from now until 11 o'clock. Oh, you know what? I think I have to give you one more. Let me see. Oh, I see. I actually spent way too much time on that. Okay, we have 15 minutes. Let me run through some other things. My apologies. Okay, so I'm going to give you an introduction to systems theory now, and we'll talk about this in a little bit more depth. We're going to review the concepts of systems theory, review the properties of living systems. We'll discuss the concept of qualitative versus quantitative qualities or properties, and determine different levels of study. This is a poem that I wrote to try to encapsulate the idea of endobiogeny. And if you can understand this poem, you can understand endobiogeny. It says, "The life that can be quantified is not life. The system measured is not the living system. The nameless is the origin of life. The named is the manager of the terrain. Those free of reductionism observe the signs of the manager. Those who see its signs follow its rhythms. Those bound to reductionism observe not the signs of the manager. Those who see not the signs follow not cause but effect. So begins all knowing." In a sense, coming to complex systems theory means going way, way back to the time of Aristotle. This is a quote from Ludwig von Bertalanffy, who is the founder of general systems theory. He was a German biologist. He says, "Aristotle's statement, 'the whole is more than the sum of its parts,' is a definition of the basic systems problem which is still valid. Aristotelian teleology was eliminated in the later development of Western sciences, but the problem contained within it, such as the order and goal-directedness of living systems, were neglected and bypassed rather than solved. Hence, the basic system is still not obsolete." When we look at a general properties of a system, we have a collection of parts which form a whole. And by nature, a living system is self-generating, cohesive. It's closed onto itself, and it's open to interaction with its environment. So here we have some opening to its environment. There's some type of metabolic processing, and then there's some type of output. And this is, in essence, what a living system is. It's self-generating, it's cohesive and coherent in its functioning, it's closed onto itself, so it's distinct from other types of organisms, but it's open to interaction with its organism on multiple levels. In fact, the nature of a living system is that it must be open to its environment if it is to survive. In general, we can say that the properties of living systems can be divided into two general groups according to James Miller in his work on living systems: energy matter and information. So energy matter has 10 different qualities: ingestion, distribution, conversion, production, storage, extrusion, movement, spatial relationships, reproduction, and boundaries. And then the information qualities are 9: external input, internal input, channeling and distribution, decoding and translation, learning and association, memory, decision-making, encoding, and output. These are the properties which exist in living systems at every fractal level of existence. And these same qualities actually can be expanded into non-living systems. So when we talk about, say, a city, or a country organization, or a transnational organization, they still share these same types of qualities. But coming back to the image that we had before, we have this concept of a system. So we have 3 different units, and each of these units has 3 different subunits. But when we look at a system this way, we really don't get a sense of what makes it dynamic in living in nature. So then we can look at a system by looking at the degrees of interrelatedness. So in this case, we see that Unit 1 is integrated in its function with Unit 3, and Unit 3 with Unit 2. There's no direct relationship between Unit 1 and 2. But what happens to Unit 1 affects Unit 3, and Unit 3 affects Unit 2. So therefore, Unit 1 affects Unit 3 indirectly. This sounds like a very abstract concept, but when we come to clinical medicine and we apply the theory of endobiogeny, if we're looking at, for example, a woman who has dysmenorrhea, we can't just look at the uterine lining and the activity of estrogen and progesterone to understand why she has excessive bleeding or insufficient bleeding or a lot of clots or a little bit of clots. We have to look at the pelvic circulation. We need to look at the role of the liver. We need to look at the functioning of the pituitary. We need to look at the activity of the adrenal cortex. We need to look at the activity of TRH, the master thyroid hormone. So even though those other factors that I mentioned are not directly linked to the uterine lining, they are indirectly linked. And so this concept of looking at complex systems and relationships is completely relevant to clinical medicine. We can expand this concept of the interrelatedness and integration of parts by defining not only that systems are linked or integrated, but by looking at if they have unidirectional or bidirectional influence. So for example, again, in a very practical clinical sense, when we look at the relationship of, say, progesterone to estrogen and testosterone, it is not enough to say that they have a relationship to each other. The specific relationship is a function of time and chronology, because at one point, progesterone supports the activity of estrogen, and at one point it starts to diminish it. And the same thing with testosterone. And there's other endocrine relationships where they're competitive, and that competitive activity could be additive. So we need to look not only at relationships, but are relationships one way? Are they two way? Are they inhibiting? Are they diminishing? Are they augmenting? These are the types of questions that we ask ourselves in endobiogeny when we look at a complex living organism like a human being. In a very simple sense, we can look at the liver as a system, and the liver is composed of multiple lobes, which would be its subsystems, and then each of those lobes contains different types of cells: hepatocytes, Kupffer cells, stellate cells, etc. And so this concept of complex systems, and then we take this one system, the liver, which has many subsystems, and we integrate it into the activity of other systems. And so these 5 organs are related to immune function. And this is not even looking at the totality of how we look at immune function. This is just looking at some simple levels of interrelatedness. We've talked about qualitative, quantitative activity. I don't know what happened there. So when we start to think in a systems way, instead of thinking on your left, if the ALT, liver enzyme, is 1,000, there's liver function dysfunction, and when the ALT is 33, there's no dysfunction, we can start to say from a systems approach that when the ALT is 1,000, there's definitely some liver dysfunction and destruction going on. But what's going on when the ALT is 33? Well, there could be a devolution in cardiac function. That's due to hepatic strain. That's due to insufficient glutathione recycling. That's affecting hepatic detoxification pathways. That leads to a relative oxidative insufficiency, which leads to impaired mitochondrial respiration, diminished ATP production, and that's due to insufficient insulin sensitivity to provide glucose to a cell as the basis of ATP production. So then all of a sudden, we're linking hepatic function in a qualitative sense. We're talking about relative dysfunction, and we're looking at how the activity of one organ is affecting the activity of other organs at the metabolic, tissue, and organ levels. So the concept, when you take a global systems approach to medicine, the idea is that you seek to modify physiology rather than control it. So this is part of this radical shift in cosmology. Instead of seeing the universe as a static and controlled phenomenon filled with predictable order, if you see it as being a chaotic and dynamic system which is integrated and interrelated and has multiple levels of influence, then your desire is not to return order and suppression of symptoms. It is to return the system to its most dynamic and functional state. Therefore, if you're going to help the organism return to dynamism rather than control and suppress symptoms, you need to have a new level of assessment which is not based on a purely quantitative level of histologic dysfunction. You need to look at how the parts affect each other. You want to look at preclinical or pre-critical imbalances. You want to anticipate illness. You want to understand what is the initiating factor, what are the entrained factors, what is the cause, what is the mechanism, and what is the result. And all of this gets turned on its head when you take an endobiogenic approach. The question is, what level of a complex system are you going to study? Are you going to study the quantum level of biology, cellular level of biology, tissues, organs, the global system, the environment, the universe? This is a quote by Leroy Hood and his team at the Institute for Systems Biology. He says, biologic information is encoded in multi-scale information hierarchy. DNA, RNA, protein, interactions, biological networks, cells, tissues, organs, individuals, and then ecologies. The important point is, he says, is that the environment impinges upon each of these levels of the hierarchy and modulates the digital information output through the genome. Thus, the systems level investigation demands the collection of data at each relevant level of the hierarchy between the phenotypic measurement and the core digital genome. So what he's saying is, are you going to study the genomic activity or are you going to study the phenomic activity? And the genome only represents the potential capability of the organism. But the phenome is the actual expressed activity, taking into account the genetic capability, epigenetic modifications, and then complex local, regional, and global systems modifications. Because remember, every cell functions for its own benefit, but not at the expense of the cells around it or the tissues, the organs, or the global system. When a system functions optimally, every level of the system functions in both modes, for itself and for the benefit of the whole. So, again, you can take a simple level of analysis, like let's say you want to look at mitochondrial function and ATP production. But the activity of the mitochondria is dependent on the state of redox, of oxidation and reduction, which produces protons through the metabolism of glucose. Okay, well, how did the glucose get in there? Well, the glucose enters because there has to be some type of signal from the membrane that the cell needs more glucose. And then there needs to be a solicitation of insulin. There needs to be a mobilization of glucose. There needs to be the proper level of fluidity of the cell membrane. There needs to be the proper phosphorylation of the internal aspect of the insulin receptor to activate it. And then we have global levels of input and output. According to the theory of endobiogeny, all of these levels need to be studied simultaneously. We don't limit ourselves in endobiogeny to any one level, because every level is significant. I'm going to skip over this. So in the endobiogenic approach, again, as I mentioned, answers are easy to come by. It's finding good questions that's the problem. And part of the challenge that we face in clinical medicine is how we conceptualize a disease, which is what we call nosology. And traditionally, we diagnose a disease based on clinical symptoms and histologic changes. So, for example, if anyone here is a rheumatologist, you diagnose juvenile rheumatoid arthritis or lupus through a series of criteria. And the patient must meet a certain number of clinical criteria and a certain number of histologic criteria, and therefore you have this disease. And if you have the symptoms of rheumatoid arthritis, but you don't meet the criteria, apparently you don't have the disease. And it sounds kind of funny to say it when you talk about it that way. But this is the current model that we base things on. And the challenge with a clinical histologic nosologic approach is that it's based on a quantitative measurement of histology, and it's based on a very subjective assessment by the patient of what exactly their symptoms are. Now, you can go into molecular nosology, which is where many scientists and researchers are going these days. But the challenges with a molecular approach to nosology and looking for single nucleotide polymorphisms or SNPs and doing genetic studies is that repeatedly, when you take people who have a disease and you analyze them for gene arrays, and you see you take all the people who are sick and you see how many of them have a gene or a number of genes and you say, oh, these genes are related to this disease. Then you do a prospective population study where you say, let's find people who have these genes and see if they develop the disease. And when you do the prospective study, you find very poor correlation. Because having the genes does not mean that you're definitely going to develop the disease. Because genes function in a certain environment. You can take an ecologic approach as well, but the approach we like is much more of a systems nosologic approach. When you take a systems approach to clinical diseases, what you see is that genes do play a role, but it is not because a gene encodes a single specific protein, but because there are common physiologic imbalances that are shared by multiple diseases. We know from our clinical medicine that people who have certain types of diseases are much more likely of developing other types of diseases. But in our current approach, we see all of those diseases as simply being separate and the result of getting a bad set of cards, if you will, in the poker game of life. But when you take a systems approach and you look at the common physiologic imbalances, you can create a map like you see here where diseases can be linked by the degree of shared physiologic imbalance. And when you start to see diseases based on common pathophysiologic activity, you don't have to have 5 drugs or 5 treatments for 5 diseases. You could have 2 treatments or 3 treatments because there are common underlying pathophysiologic activity. So one of the benefits of a systems approach is that it lets you simplify your treatment based on treating physiology rather than treating symptoms or a disease state as we've diagnosed it. So a systems biology approach says, how does the disease develop? But a global systems approach, like endobiogeny, says, why did the disease develop in this person? And to us, that is the more pertinent question. So the question I will leave you with until after our break is, what is the system and what manages the global system? Because if we want to study the system, we have to understand what manages the system and manage the manager. So I'll leave you with a quote from The Matrix, from those of you who've seen this movie. The Matrix, for those of you who don't know it, is about a man named Neo who discovers that that the world that he lives in and the whole belief structure is false and is being propagated by people that want to keep him from knowing the truth. And he meets a man named Morpheus who offers him a chance to either take a pill so he can see the truth, or take another pill and go back to doing the things that he used to do. And so Morpheus says, this is your last chance. After this, there's no turning back. And I paraphrase to put this in this context of medicine. You take the blue pill, the story ends. You wake up in your clinic and you practice medicine the way that you want to practice it. You take the red pill, you stay in the fellowship, and I show you how deep endobiogeny goes. Okay. So we'll take a 15-minute break. We'll be upstairs, yes, on the 6th floor. We'll have a 15-minute break, then we'll come back and talk about the